If you are a NFL fan, then it's Super Bowl week. Um, the it's the Eagles and the Chiefs. Got to be honest with you, I'm a Green Bay Packers fan. And so I kind of want both teams to lose. And that's not just me being a spoil sport. I don't care for either of those teams. At any rate, um, today what we are going to do. Oh, let me reverse for a second. Um, I am going to be finishing your practice exam. I said that I was going to get it on Friday, and I'm sorry I did not. I will be posting your practice exam before the end of the day tomorrow. I have to catch up on some grading, so you're going to be seeing some responses and some grades back in the next few days. Um, we have an exam, I believe your class is synced up with my other class or one of my other morning class. Yes, our exam is going to be on Wednesday, the 12th. So that's a week from Wednesday. Um, so to prepare for your exam, you'll have the practice exam. You'll also like on Wednesday, that will be the end of new material. And so a week from today in class, so on February 10th, um, we will be taking our exam in class. Nope, 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 no, no, no. A week from today, February 10th, we will be reviewing for your exam. So before that class, you'll have a good number of and good resources to review for. I'm trying to figure out how, and I'm sure that it's easy to do, but I'm trying to figure out how to export all of the poll questions that I posed. And then there's a couple of poll questions that I have not asked, but I want to give you those so you again see like how does Joel write questions? What do his questions look like? You have two quizzes that are up and yep, three quizzes that are up and two assignments. One thing that I did different this week from last week was I've added like a very short description about where the material on the quiz is coming from. So, for instance, if you look at quiz number, I think it's nine, no, or 10, the effectively the first quiz of this week, you'll see a description that most of the content on that quiz is actually coming from last Wednesday's lecture. Just some more questions related to that. And so I've got those brief descriptions. So that will give you an idea of like if you want to, if we haven't talked about the stuff, I wouldn't start taking the quiz yet. Um, and if you feel more comfortable as I post the PowerPoint for Wednesday, which should be up later today, then by all means go for it. But if you, yeah, just it will give you a better idea of when you should kind of take that quiz. Um, Great question, Brooks. So your exam is going to be mostly multiple choice. There are going to be some that are multiple answer. Just kind of an idea of the exam. If it's roughly 40 questions, that's what I'm aiming for. Then about five of them are going to be multiple select. And what I mean by that is, the question stem will be something like select the true statements from those below. And like two of them, maybe three of them will be correct. The activities do not. I'm sorry about that. Thank you for bringing that to my attention, Stanley. Um, the activities I need to add the description. So they are up. If you go to our homepage, if you go to the activities column on our homepage, you'll see that the links are there. I'm taking a moment to go ahead and add those links so that they're good. Let me do that right now. I'm sorry, I posted them, but I didn't direct the link there. So let me do that right now. Activity 5. I'm going to save this and then activity 6. And when you go back to those descriptions, you'll see that in there. Sorry about that, everyone. Um, then save. Okay, then last but not least, I need to do one more thing. Um, okay, so activities are available and up, they're good to go. Quizzes are available. Like I said, if you want to take your quiz for, or if you want to do them all right now, then kind of do so at your own, well, just I haven't talked about them yet. So, or I haven't talked about all the material yet. So just be kind of aware of that. Then, this is what I want to delete that file. And I did make a mistake on the PowerPoint that I uploaded. Um, and I just saw it a moment ago as I was kind of readying for class. I will point this error out for you so that you can make that adjustment. So today, what we are going to begin is we're going to start talking about amino acids. And amino acids are a class of compounds that actually, go ahead and add to the chat if anyone, have you ever heard of an amino acid? And if you're maybe you've heard of them in the context of BCAAs. Has anyone ever heard of a BCAA? Protein, perfect. Has anyone ever heard of a, yep, protein shakes? BCAAs, if you've heard of those are also known as branched chain amino acids. Lots of people take those as a part of like a pre-workout supplement. Um, and branched chain amino acids, one of the things that they're kind of supposed to do is um, help kind of get your blood flowing a little bit, give you a little boost of energy, kind of like B vitamins. Most of the time, whenever I talk about or if I've ever mentioned a pre-workout supplement, pre-workout supplements have lots of B vitamins and caffeine. Caffeine, they got a lot of caffeine. Sometimes they'll also have an amino acid known as arginine. But we're going to talk about, and we're going to start talking about amino acids in the context of they're the building blocks for proteins. And that phrase right there, building blocks of proteins, is one that I think is going to be really helpful for you to kind of keep in mind. Now, I'd like to go ahead and give you guys a first poll question for kind of organization and to think about how we classify amino acids. I can go ahead and launch this one. What indicates that a molecule is polar? Kind of what's the best indicator? Is it a carbon, a hydrogen, or a lone pair of electrons? So whenever we talk about amino acids, we generally classify them in basically three different major groups. They're polar, they can, under that kind of umbrella, they can be positive, they can be negative. And then there's one last category of polar amino acids, and that is known as uncharged. We're at about 90 or 85% of the folks in class responding. We've got of these different options, what's the best indicator that a molecule is polar? Yeah, it's a lone pair of electrons. You're absolutely right. Um, a hydrogen, yeah, you're going to find that on a polar molecule, or you potentially will find it on a polar molecule, but you'll also find it on a non-polar molecule. So between these different answer options, a lone pair of electrons is kind of going to be your best answer. Now, um, amino acids are going to fall into these different major categories: polar, non-polar, and aromatic. With respect to a polar, or this class, polar, um, a polar molecule is a molecule with a charge or lone pairs of electrons. It's a molecule also that's not symmetrical. Now, I know that whenever I think about symmetry or something that's asymmetrical, or go ahead and throw in a definition. What's symmetry, or what does symmetry mean? Reflecting images, yeah. Same on each side. So one of the things whenever we talk about symmetry is we always have to have like a plane. So, or a kind of something that divides it. So if you took a line and drew it straight down my face, I know that I read about this. When I think of nonpolar, I think of oh, okay, so nonpolar things generally are going to be symmetrical. So yeah, yeah, great, great. That's awesome. That's awesome. I love that. Nonpolar, normal, symmetry. Awesome. Because they kind of balance one another out. Now, what I was saying was, I know that my face is not symmetrical. I know that I heard this I feel like I saw it in Newsweek or Time Magazine or something like that. And it was like, um, beauty is related to facial symmetry or something like that. And I was like, that's it. That's why I'm not a model, I guess, or a face model. At any rate, you got something that's absolutely identical on each side. Now, when we have these different molecules, we classify them as positively charged, negatively charged, or uncharged. The kind of opposite of nonpolar would be, or sorry, the opposite of polar would be nonpolar. Nonpolar compounds are molecules or compounds that have no charge and they have no lone pairs of electrons. A good example of a nonpolar molecule would be something like a hydrocarbon. So something that's just carbons and hydrogens. Because, carbons, as long as they're, as long as a carbon is making four bonds, it's in great shape. Now, our next class that I want to talk about are, are aromatic compounds. When we talked about aromatic compounds, could anyone add to the chat? First of all, it's a molecule with a ring, but then there was one other criteria or one other kind of detail about an aromatic compound that we were looking for. Could anyone add that to the chat? What the kind of second level of alternating double bonds? Absolutely right. So if you've got a ringed molecule with alternating double bonds, then you're in good shape. You've got an aromatic compound. So this is well, it's a six-membered ring. So right now I've drawn a hydrocarbon, but then whenever I put that in, well, whenever I add that double bond, what suffix do I need to add now that I've added a single double bond? Close. The NE is right. ENE, perfect. So that ENE suffix indicates that I've got a double bond. And so this would be cyclohexene. Now I've got two double bonds. My suffix stays the same. It's still ene, but I have to indicate, 1,3 di or cyclodihexene. But then whenever I add my third double bond, now I've got double, single, double, single, double, single. So I've got alternating double bonds. This is an aromatic ring, but I know you guys are probably more comfortable with and probably like drawing it like that a little bit more. It's kind of a hexagonal donut of sorts. Now, we're on to the compounds that we're interested in. I'm jumping right into these because these molecules are going to be important. These molecules are the building blocks for proteins. And the first thing that I want to introduce to you is the names. Now, I'll add a little bit of a detail. We're going to refer to these as the protein ogenic. Now, this means that, or that term proteinogenic, that means that they're protein builders. These are the molecules that you assemble together to make a protein. You might also see these referred to as alpha amino acids. And I'm going to add a little bit of detail, and that is a proteinogenic means used to assemble protein. I'm going to say proteins. Alpha amino acids, they have an alpha carbon. Hmm. I'll show you that in just a little bit. An alpha carbon. And yeah, those are the important parts about and important terms that you might hear amino acids referred to as. Now, I bring this up because, well, an amino acid needs two functional groups, an amino group and a carboxylic acid. So, an amine and a carboxylic acid. There are tons of different molecules that have an amine and a carboxylic acid. Does that mean they are all amino acids if they have those two things? It kind of does. But does that mean that they are all, and I'd like you to respond to this, I'm going to lead you a little bit on this one. Are all amino acids used to assemble proteins? I like it. Probably not. You're absolutely right. Not all amino acids are the building blocks for proteins. Only a select number of them. And I'm going to share with you right now the 20 names of our proteinogenic amino acids. Now, what do you notice about most of these molecules and most of their names? Is there something in common that you see over and over again? The ine suffix, you're absolutely right. Not every single one of them, but I mean, heck, if I count them up pretty quickly, got my acids and fan. If I've got 20 of them, about I'll call it 17 of them have that ine suffix. That ine suffix, where do you think that originates from? What do you think that's kind of calling back to? Probably the amine. Okay, so these names, these are the 20 different names. We're going to look at all of these structures, some of them today, some of them on Wednesday. On your exam, do I want you to know all of these names? I don't want you to know all of these names. What I want you to be able to do, however, is identify a couple of them. And I'm going to specify the ones today and then specify some other ones on Wednesday. Your activities for this week do have you drawing them and drawing a couple of these structures. And they also have you um, looking at the codes that are used for them. So our amino acids, our proteinogenic amino acids, they have these full names, but they also have what are known as single letter and three-letter identifiers. Okay, so I'll give you a little bit of a clue. The ones that I want you to know are these two: lysine, histidine, and arginine. So those five amino acids are the ones that I want you to know the names of. And I would say that those are going to be good ones to kind of reference more times than not. I select these two, the aspartic acid and glutamic acid, because these are polar negative amino acids. Lysine, histidine, and arginine are polar positive. The remainder of them are classified as either polar uncharged or nonpolar. If you're thinking to yourself, well, what's something I need to do today after this class? I would say make sure that you know the full names, be able to translate between the full name of these five amino acids and their three-letter codes, and their three-letter codes and their one-letter codes. So think about it almost as like if I had a short answer question, which you're not going to have short answer exam questions on, or you're not going to have short answer questions on your exam, but I'm thinking about the depth and comfort that you need. Well, these are the five that you kind of want to dive the deepest on. The others we'll get familiar with, and I'll give you a little kind of almost trivial factoids that you need to know about them. But those are the ones that I want to get you started with. Now, like I said, all of these amino acids have full names. They also have three-letter identifiers. And these three-letter identifiers go back to a field of biological research known as bioinformatics. Bioinformatics. Add to the chat, what do you think bioinformatics means or relates to? Let's say that it's the biology of or studying biology using. What do you think? Add a term there. Or add, okay, I like it. DNA, absolutely. So bioinformatics is a field where basically you're using huge amounts of data, whether that's DNA analysis or protein analysis, but you're doing it with really intense calculations in computers. And this, there's a woman by the name of Margaret Oakley Dayhoff, Margaret Oakley Dayhoff, who was a bioinformaticist back in the 1950s. And she was, working on computers. She was basically she was analyzing proteins and she was like, typing out the full names when we have a protein that's made up of 500 amino acids, typing out all of these names, it gets really hairy. And if you have a typo, then it screws up your calculation. So she was like, we've got to do something better. And so the work that she did led to the development of these three-letter codes. Now, here I want you to see all of the three-letter codes, and I want you to, while you're looking at this, answer this poll question. How many amino acids do you notice do not have the same first three letters in their name as their three-letter code? So I'll kind of lead you along with this one and say, well, if we look at aspartic acid, the first three letters are ASP. Serine, SER. Cysteine, it's the same. So I'm just going to put an X if it's the same. And then I'm going to start a tally if they're different. Leucine is the same. Glutamic acid, threonine, proline. Ooh, isoleucine gets a tally. Lysine, same. Asparagine, ASN, as opposed to ASP. Okay, so I got a tally there. Valine, VAL, same. Tryptophan. Now, if you downloaded the PowerPoint or the PDF like 10 minutes before class started, well, then this one actually has a typo. That's why I replaced that file. So this tryptophan is TRP. So I'm going to add my tally up there. Histidine, HIS. Glutamine gets a tally. Glycine, same. Phenylalanine, same. Arginine, tyrosine, alanine, methionine. We got four that have, well, differences. Now, looking at those, would anyone care to add to the chat why? And while you're adding that, I'm going to end the poll. I got 26 out of 29 respondents. 65% of y'all said four. So 65% of y'all were right. Okay, now let's check the responses. There would be dual names. Absolutely. Now, if you look at aspartic acid versus asparagine, that's an instance where you have an issue. Now, some of these three-letter codes, well, that's where we have a conflict. Tryptophan, I mean, you could do TRY, but they elected to not do that. I think maybe it's just a matter of, well, TRY, TYR that could easily be mixed up. Then glutamine, GLN. Well, glutamine and glutamic acid. Okay, glutamic acid and aspartic acid. This is kind of interesting. Then what was our last one? Isoleucine, ILE. Okay, so we've got glutamine and glutamic acid, asparagine and aspartic acid, tryptophan and tyrosine. If we did TYR, that might be a problem. Um, that's a great observation, Alex. It gives you a little bit of an insight into the suffix, but also these different names contribute to the structures. And I'm going to show you those structures. And since we're really focusing on basically only five of our amino acids, I hope that this will be really helpful. In just a minute, we're going to get to the structures. Um, but one of the reasons that isoleucine gets ILE as opposed to ISO is, have we seen that prefix ISO before? And if so, what did we see it in? Spoiler, yes, we have. An isomer. An isomer is a very, very commonly used term in chemistry. And so it's almost like ISO was taken. So you got to think of something else. And that's where ILE came from. So there was a paper that was published back in the 1960s about the assigning of these three-letter codes. And the process was, well, let's give every single amino acid the first three letters, unless that's taken. In which case, let's pivot, let's do something a little bit different. Okay, so in addition to the three-letter codes, we also put together one-letter identifiers. And we see with those one-letter identifiers, I'm going to give you another poll question. How many amino acids have single letter codes that are not aligned with or not the same as their first letter of their name? In this poll, I've got 0 through, I think, 10. So we got quite a few different options there. I'm going to go through these with you guys. So ALA, same. Cysteine, same. Aspartic acid, I'm going to start my tally. Different. Glutamic acid, different. Phenylalanine, yeah. Bonics, F, but F and P aren't the same, so I'm going to do that. GLY, HIS, ILE, LYS for K, or lysine gets a K. LEU, LMET, ASN for done. PRO, same. Q, glutamine, different. ARG, arginine, different. Serine, same. Threonine, same. Valine, same. Tryptophan, different. Add that on there. Last but not least, tyrosine gets a Y, so we're at a grand total of 9. Now, we had 4 three-letter codes that differed from the names, but then we got 9 one-letter codes that differ. Now, with that in mind, Andrea pointed out that we'd have dual names, dual three-letter codes. You're absolutely right. And in here, if we go with alanine getting the A, well, then that throws off aspartic acid, which also throws off arginine, which also throws off um, asparagine. Now, that was kind of the rationale. That was the approach that was used. Okay, I'm gonna go ahead and end the poll. We got to 93% of the folks responded, and 63% of y'all selected the right answer of 17, or sorry, of 9. Um, Now, with that in mind, though, let's look at some of those single letter codes. Are there any single letter codes that, as you say the full name, um, anything resonates with you? The one that I always like to think about is arginine. Does anyone want to guess what a pirate's favorite amino acid is? Marcus, great question. Um, Arginine is a pirate's first amino acid. Are we going to have to memorize all the identifiers? What I want you to do is be able to go back and forth between single letter and three-letter codes. So, what to answer your question, Marcus, I feel comfortable saying no, because what we are focused in on is we are focused in on these first five amino acids are polar positive residues. Now, for your worksheets and for your activities this week, yes, it will be important to be able to toggle back and forth between them. And you will kind of need to recall those, but you have those on your worksheet. Um, or you have access to this. Now, another thing that I wanted to add is that when you take your exam, um, you guys can use a 3x5 index card. So you can write notes down like this. Um, so now I'm going to go back over here. So, like I said, a pirate's favorite amino acid is arginine. And I know that whenever I was in kindergarten and learning how to read, some kids learned how to read using, I'll give you a hint. It started with a PH and then it ended with onyx. Did everyone have any experience with, like, Hooked on Phonics was popular whenever I was a little kid, and then people got t-shirts that said it, and it was silly, but, um, like the phonetic pronunciation of things. You look at the word and the way that you pronounce it, um, or the way that you read it relates to the sound that those letters make in combination. So arginine is an example of a phonetic pronunciation. Aspartic acid also has kind of that phonics designation. So aspartic, well, T is already taken. So it's almost like this was pronounced aspartic. So that's basically how it was assigned. So essentially, whenever it came to the single letter identifiers, um, there was kind of this set of rules and guidelines. The first thing that we're going to do is we're going to say, well, assign first letter as single letter, but then with a little bit of a detail, unless already used. Okay. And then that brings us to our second point. Well, how do we assign them after that? You use the phonic or phonetic, I think is the proper word, the phonetic pronunciation. And if that doesn't work, for instance, glutamic acid getting an E, why? What the what the scientists did with this was they looked at structure of the molecule. Molecule. Is this whole table of amino acids or there? Oh, that's a great question, Alex. Um, there are other amino acids. These are all of our proteinogenic amino acids. So these are the ones that are used for synthesis or used for putting proteins together. Um, or the ones that is this whole table of amino acids or all there are? No, or the ones that get these codes. Sorry, I just had to process your second question. There are other single letter identifiers for other proteinogenic amino acids. Do non-proteinogenic amino acids get codes too? I don't believe that they do. That's a fantastic question. You guys are asking awesome questions. Um, so there are, if you think about like every living organism. And what I mean by that is like if you think about a person and then you think about a dog or you think about bacteria that live under Antarctica, there are some amino acids that are used to make proteins in my body and in your bodies that are not, or sorry, and then there are some amino acids that are different in these microorganisms that live under Antarctica that are used to make their proteins. So there are other proteinogenic amino acids, but these 20 are the main ones. And the two or three other ones, they do have single letter identifiers. But those would be like the B and the O, get those. Um, at some point, though, you run into the problem of, well, we only have 26 letters in the English alphabet. Um, how else would we identify them? But then, if you think about like, well, every single protein in your body, every single protein in a piece of steak, well, all of those proteins are made up only of these molecules, only of these codes. So you guys are asking fantastic questions, and thank you very much. And I hope that I'm answering them, I hope that I'm answering the correct questions that you're asking. If there's something that is unclear in my response, please don't hesitate to either say something or ask a follow-up question. So, so we've got this process where we look at the first letter of the name, and then we assign that first letter as the single letter code. But if we have a redundancy, or if we have like a duplication, like aspartic acid or arginine, well, we can't use A for those. Well, then the idea is and then, it's not, okay, then let's go to the next letter because, well, the next letter in aspartic acid is an S, but serine's already taking that. So that's when we go to this phonetic pronunciation. And we say, basically, how do English speaking people say that word? And is there like a strong letter that you pronounce when you're saying that word? And so if you think about phenylalanine, that uses an F. Think back to when you were young and you were learning how to spell. And I see this with my kids. If they are spelling the word phone, they will start with F. And then they'll correct it and PH. But that's where that phonetic pronunciation comes from. If the phonetic pronunciation doesn't address it, which it does for aspartic acid, phenylalanine, and arginine, then is there something else? Well, that's whenever people looked at the structure of those molecules. And so to illustrate that, I want to say, okay, well, aspartic acid and glutamic acid, I want you to think about those two amino acids because I want you guys to know the full name, single letter, and three-letter codes. Awesome. Awesome. The single letter, full name, and three-letter codes. I want you to go back and forth between those. I also want you to be able to identify those structures. Today, I'm going to show you our polar amino acids. I want you to be able to identify the structures of the polar positive and polar negative amino acids. Does that mean you have to know every polar structure? Nope. There's a total of 5 structures that I want you to, whenever I show it to you on an exam, I want you to be able to identify it. So, with that in mind, what I'm going to do is I'm going to jump ahead. My next slide is polar positive, but I'm going to jump ahead to polar negative. Okay, so boom, boom. Here we go. Here are our polar negative amino acids. Now, what I want to show you is this is both of them. This is aspartic acid and glutamic acid. Aspartic acid has the single letter code of D. Glutamic acid has the single letter code of E. Now, when we talk about classifying amino acids, there are some things that we kind of take for granted. So, does anyone recognize a sequence of atoms that is identical between these two structures? And or do you notice anything that you're like, oh, well, that's the same in both of these molecules? Chris, please stop reading my mind because that's exactly what I was going for. Okay, carbon to the oxygen bonds, great. What I'm going to circle right now, and you know what? Here's one of the things that's tricky about my ability to use PowerPoint is I think that I can zoom in. Yeah, there we go. But now I think I lose my drawing ability. Yeah, I do. But never mind, I've got it figured out because I can use, okay, I'm going to zoom in. That's good right there. Now I can use my annotate function with my non-PowerPoint drawing. Okay, so, and that one goes over there. So if I draw, let's see, just a second. I hope I don't go to the next screen. Nope, I didn't. Okay, draw, pen. Nope, that's not working. Okay, just a second. Sorry, everyone. Bear with me for a second, and I'm going to get it sorted out. But now I got to zoom back in, annotate tool. Okay, pen, draw. Good. Okay, so what I'm going to do is I'm going to circle. Oh, I can use white because, yeah, right here. This, what I've circled, is known commonly as the backbone. So that backbone is kind of the thing that every single amino acid has, and it's the exact same. So what makes every amino acid different from one another is the part that comes off of the backbone. Now, when you look at, and this part that comes off of the backbone is commonly referred to as the R group. Now, if you think back to when we talked about organic chemistry, did we have kind of well, a definition almost for an R group? Did we talk about R groups? Alex, absolutely. Everything else. What makes this different? What makes amino acids different? Yes, Chris, absolutely. Many times those R groups are just long hydrocarbon chains. When we talk about an R group in the context, or sorry, let me rephrase that. When we talk about R groups and we are talking about amino acids, we're talking about the part that matters, the part that's interesting. So it kind of takes you to like flip the script and flip the way that you're thinking about it. Now, when you look at those two R groups, is there a functional group that you recognize on those two R groups for aspartic acid and glutamic acid? A carbonyl. Absolutely. There's a carbonyl carbon, and a carbonyl carbon is found in a handful of different molecules, a handful of different functional groups. I'm going to extend this and say this whole thing right here. We maybe, I know that I referred to it as this. All right, let me rephrase it. COOH. What does that COOH mean? And maybe you saw me draw it like this. It's always weird when you're looking at it in different axes. Carboxylic acid. Okay. Now let's piece some things together. Both of these molecules have a carboxylic acid, right? Give me a thumbs up or a yes in the chat if you agree with that. Circle them. Do these both have a carboxylic acid? Yeah. Okay. Now look at the name. Aspartic acid, glutamic acid. That suffix or that additional word, or actually that suffix, the IC acid, ic acid, is where this is coming from. What was the name you gave at the top half? Oh, Let me write that out. Thank you, Chris. Backbone. It's a B. So there's our backbone. Yes, Taylor. These are, let me add that in. I'm going to thank you very much for adding polar negative. These are our only polar negative amino acids. So our polar negative amino acids are aspartic acid and glutamic acid. Now, I got to be honest with you guys. I feel like I learn best whenever I can draw like a, or whenever I can make a parallel or a connection to something else and or like tell myself a story. So it is the ick at the end that are using. Yes. Do we get a version of the slide with your annotations? I got a question. Do we get a version of the slide with your annotations? Um, for the most part, yes. These, what I'm going to do right here is the these markings that I'm making right here will not be translated, but I'm going to do a screenshot and then oh, wait, no, no, no. Screenshot. I'm going to grab all this and I'm going to throw it into a Word doc so that I can post that on our Canvas page separately. Um, it just has to do with the different, like whenever I zoom in and I try to write on the screen, PowerPoint doesn't like that. Um, and so I don't get to. But I've taken a screenshot, I've added this. And so since I can't write on it with PowerPoint, I have to use yes, I did that, Javier. So yes, snipping tool is fantastic. It makes me feel like a genius because I'm like, oh, I just want that page, that part of the screen. Um, okay. So with all that in mind, okay, these are our two polar amino acids. The R groups both have a carboxylic acid. Let me erase some of what I've written. And okay, so these are our two R groups that have carboxylic acids. Um, then what I want you to notice also, I'm gonna erase those carboxylic acids that I drew. Like I was saying, I always like to assign or give a story to these molecules. That helps me learn them, remember them, memorize them, whatever. Whenever I look at these two, aspartic acid and glutamic acid, I see that they both have a carboxylic acid, but then the other thing that I notice is this right here. The R group for aspartic acid is basically CH2. The R group for glutamic acid, after, or sorry, taking into consideration the carboxylic acid, is CH2, and then another CH2. So it's like CH2, CH2, carboxylic acid. Does add to the chat if you follow me there. Okay. So I look at these two, and the story that I kind of assign to them is, well, structurally, they're really pretty similar. And I always kind of think about them as being, this is the maybe weird personifying part, is I think, well, they're siblings. They're related. Aspartic acid is the shorter sibling. Glutamic acid is the taller sibling, which is why it has the extra CH2. That's also helpful, and this is maybe where it also goes a little weird and off the rails, is if I think about the alphabet, which one comes first? D, then E. So we have this sequence, D and E. That's the sequence in the alphabet, and then I always just think like, well, the shorter one gets the first letter, and then the taller one gets the second letter. So that's why basically the D Single letter code was assigned to aspartic acid because of the phonetic pronunciation of aspartic acid. And that might be a little bit of a stretch for some folks, but that was kind of the rationale. That was the decision. Then glutamic acid. Why did it get an E? There's not an E in the name glutamic acid. Well, it's because if you look at the sequence, I like it. Glutamic looks like an E. Yeah, works. Um, it's because, well, they're so similar to one another that at very least we can put their letter sequence together. Okay, so what we've gotten after here and what we've looked at here is we've identified a couple of things. The amino acid backbone. Every single amino acid, every single proteinogenic amino acid has this same backbone. And if you look at that backbone, and I think I've got black. We've got a carboxylic acid. We've got an amine. Now, if you remember, I referred to these amino acids, or I referred to the amino acids as proteinogenic amino acids. What was the other term that I used to describe the amino acids? I'll give you a hint. It had to do with alpha, alpha, alpha amino acids. Perfect. This carbon right here, I'm drawing a circle around it, and this carbon right here. What do you think that carbon is referred to as or known as? The alpha carbon. Absolutely. Perfect. Thank you so much. You guys are doing awesome. This is the alpha carbon because the R group comes off of that alpha carbon. Now, what's really helpful for me whenever I draw an amino acid is I think of the pattern NCC. NCC. Every single amino acid is going to have that backbone. Then I know, where does my R group go? Well, it comes off of a carbon. Well, I know that this right here is known as the carboxy termini. And that's where my carboxylic acid goes. This right here is the amino termini. And that term termini, meaning like terminal or end, the amino end or the carboxy end. And that amino end, I'm going to say NH3 plus, which leaves my alpha carbon. My alpha carbon, I put my hydrogen on there, and then I put my R group that comes off of that. Now I'm going to erase that R group because one of the things that gets a little bit challenging is what is the single letter code for arginine? R. So it can get kind of hairy whenever you're like, wait, the R group for the amino acid R. Oh, rats. You're kind of using a redundant term there. Okay. Now, before I zoom out of this, I want you to know how to be able to identify these two structures. I want you to, on your exam, if I gave you the structure of aspartic acid and it was unlabeled, I want you to be able to say that this is aspartic acid, or this is single letter code D, or this is ASP. I want you to know the full name, single letter, and three-letter code for these two molecules. And then the other ones are polar positive residues, which we're going to go to in just a second. Okay, so I got a screenshot of that, snapped it into Word. I'm going to update that document or upload that document after class. I'm going to, if anyone wants to take a snapshot of this while we're here, I'm going to give you just a little bit of time to do that before I erase everything. And before I go back, does anyone have any other questions related to our polar negative residues? And I'm going to mentally count up to about 15 so that you guys can have some time to ask. Okay, I'm going to clear all my drawings. All drawings. Okay, there we go. Then I'm going to zoom back out. Oh, too far, too far, too far, too far. Those were our polar negative residues. Now we're going to look at our polar positive residues. Our polar positive residues are lysine, histidine, and arginine. The I'll zoom in again for this. And this is again an instance where I'm gonna have to grab a screenshot because of oh, my drawing limitations there. Let's see, can I do that? Okay. I have to minimize the chat for just a no. Okay. No, I can move it around. I'm going to start out with lysine. Actually, I'm going to start out with all of them and go with my backbone. I want to circle my backbone, and then I want to indicate my alpha carbon. There's my alpha. Next backbone. Alpha. And then arginine, last but not least, alpha. So these are three more of the total of five structures that I want you to know. Lysine has a single letter code of K and it's a three-letter code of LYS. Histidine was fortunate. H wasn't taken by anything else and has the three-letter code of HIS. Arginine, well, we had to go on to R for that phonetic pronunciation and ARG. Now, lysine is kind of a tricky one. And I think that if we think about the process for identifying and giving these different codes, um, nobody pronounces lysine choline. Similarly, it doesn't really have a obvious K structure or anything like that. Lysine has the letter K because, well, K wasn't used by anything else. Because if you think about INE, the full name lysine, every single letter in the full name of lysine is a single letter code identifier for another amino acid somewhere in that molecule. So we had to go with K because it was unused. Okay. Now, the structure for lysine. I want you to know the structure, but what I want you to know more than anything else is that at the end of lysine, we've got what type of functional group? Or, sorry, what functional group do we have there? We've got an amine. So whenever you're thinking like, how do I identify this amino acid? What do I need to do? I would like you to just say, okay, it's an amine. And so if you're like, oh, how am I going to remember lysine has one, two, three, four carbons off of the alpha carbon? I'm not going to lose any sleep over that. I want you to look at lysine and just think backbone and then CH2, or there's some carbons, and then that amine. It has an amine functional group on its R group. Histidine. Well, histidine here, this is a ring. It's a five-membered ring, no less. What I want you to take home about histidine is it's got a positive charge. It's a positively charged ring. So if you're thinking, what kind of questions might he ask about identifying amino acids? I could say, Jeopardy style, this amino acid has a positively charged ring. And then your answer would be, what is histidine? Then, last but not least, arginine. Arginine and lysine, I struggled to keep those straight for the longest time. But what became very helpful for me was whenever I realized, well, arginine has a couple of nitrogens. Lysine has one nitrogen. They are both positively charged, which is why they're classified as polar positive residues. So if you're thinking to yourself, okay, I've got to remember these five structures, where should I start? My first recommendation is to start with the two polar negative residues because there's only two of them. Look at those structures, learn those structures, learn those single and three-letter code structure of those structures. After that, go on to the polar positive residues. And with those, just think, okay, one of them has three nitrogens, one of them has a ring, one of them has one nitrogen. You can kind of simplify it for yourself like that. So these are our polar positive residues. I'm going to go ahead and minimize the chat for just a second so I can get a snapshot of this with kind of those clue recommendations for how I would suggest taking that on. Now, does anyone have any questions about these amino acids before I clear my drawings and then go on to the last part of today? And I'm opening the chat back up. Okay, cool. My last amino acids, clear all my drawings because they don't all, wait, how do you remember arginine? Oh, arginine, three nitrogens. That's what I do. Three nitrogens and a positive. Absolutely. Okay, now I'm going to clear my drawings. Okay. This last group of amino acids are polar uncharged residues. Serine, threonine, asparagine, glutamine, cysteine, tyrosine. There's a total of six of them. And what you need to know about these, and what's most important for you to know about these is not the structure, but instead the types of interactions. So I'm going to say that polar residues, actually, here, let me erase this. I don't know if anyone watches college football. I do from time to time, and they always I watch I watch a lot of SEC football, and they always scan the crowd and they show people and there's always people wearing like buttons that say, my organization hearts Bama football. So I'm going to say polar amino acids heart hydrogen bonding. So if Syrian went to a football game, it would wear a button that said, you know, serine hearts hydrogen bonding. Roll Tide. Yep. Yep. Oh man, actually, oh no. Ooh. I gotta be honest, I kind of like that. I can't remember who the commercial is, which indicates the commercial didn't work. The one where the guy's like, high tide, or what is it? Is that a Geico commercial? It's probably not. I don't know who it is. Okay, thank you. Thank you. I like that one just because I imagine going and being like, high tide, and then all the players being very confused. Um, so what I want you to take away from your polar amino acids is I want you to know their classification. Um, and what I want you to recognize about these, I don't want you to commit the names, the three-letter or the one-letter codes to memory. Instead, what I would like you to do is if I showed you one of these molecules, I'd like you to be able to identify the group that makes it polar. And what I mean by that is if you look at a serine right here, what makes this amino acid polar has nothing to do with the backbone because every single amino acid has that backbone. What makes this amino acid polar? Does anyone want to guess? You're looking at the R group. Chris, I told you stop reading my mind. You're absolutely right. Asparagine. Well, this whole thing is polar. Glutamine, this whole thing is polar. Threonine, polar. The captioning is over where I would otherwise draw. But the reason that I say that is because one of the questions that I always like to ask is I like to give a question and I'll say, hey, a new amino acid has been discovered. Here's the backbone. And here is what the R group looks like. I had this professor as a graduate student who would ask these questions where it was like, you've traveled to a distant planet and you've discovered this. Absolutely, Alex. What we're looking for to identify something as polar is we're looking for like an OH group. The reason that we're looking for an OH group or an NH is because if we look at OH, an alcohol, here, I'm going to zoom in a little bit, or I'm going to draw what serene's R group looks like. We've got that OH. Do you see my translation of this R group right here? I'm translating the R group to that right there. Okay. So if you can see the kind of connection there, what is not drawn in on that is these two lone pairs of electrons. Those two pairs of electrons make that molecule polar because what they do, and so sorry, Alex, yes, that is something I did not include on this image, but in just a second, I'm going to complete that question that I've got over here on the left-hand side. Whenever you see an oxygen and you see it with, like here, it has a bond going to hydrogen and then a bond going to carbon. And I know that I just drew over that, so it's hard to see. What you also have are these two lone pairs of electrons. And what those two lone pairs of electrons do is they're like, well, they make this area right here, where I just drew that little star, a little bit more negative. Does that help, Alex? So, and if you have follow-up questions, by all means. I knew about the... Gotcha. Gotcha. So, anytime you basically a good kind of trigger or clue for you should be OH or NH. If I see that, I'm thinking polar. So on the left-hand side of the screen here, one of the questions that I love to ask would be, and actually, heck, let's go ahead and do it like this. See, and this is kind of a negative question. When you look at this molecule, I want you to be able to see, and I'm going to draw on the exam, I'm not going to do this, but I want you to be able to look at this molecule and think, okay, backbone, done. Alpha carbon. Now, what comes off of my alpha carbon is my blank group, my R group. Perfect. Okay. So when we talk about classifying amino acids, are we talking about the backbone? No, we are just talking about the R group. Now, when you look at that R group, if you see an OH or an N, that could be an NH, that could be an NH2, or it could be an NH3. Basically, if you see a nitrogen, I want you to think it's probably polar. I'm not putting $1,000,000 down, but I'll put $20 down that it's polar. Now, looking at this molecule that I just drew, this is a fake amino acid. My sort of negative question is, would you classify this as polar? Yes or no? You would not. Fantastic. Is there an OH on that R group? Nope. Is there an NH, an NH2, an NH3? None of those are there. So that means that if you don't classify it as polar, what would you classify this amino acid as? Close. It's got lots of covalent bonds, but it's either polar or it's non-polar. Wonderful. Great response. Great, 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 great. Okay. So I've given you the five amino acids that I want you to know the structures, three-letter and one-letter codes for, and their full names. But the questions that I like to ask about amino acids aren't so much, did you commit this to memory? But instead, hey, here's a molecule, apply what you know. And this right here, classifying this as a non-polar amino acid, is applying what you know. So I'm going to erase, and I'll give you another one. C. And then we'll go group by group. Okay. So where I'm drawing a number one, this is a functional group. What is that functional group that I just drew a kind of box around in red? A carboxylic acid. Fantastic, Chris. That carboxylic acid, if we think back, I'm going to toggle back, jump back. Oh man, we saw a carboxylic acid on our polar negative residues. Stanley, that's a great question. I think that I'll go ahead and put a box around the backbone just to be absolutely clear. This is what I'm asking, or this is what this is what I'm asking about, or this is what I'm asking about. So I will indicate, this part is the backbone. Thank you for asking that. Okay, so now in this molecule that I just drew, I put a box around a carboxylic acid. The red box is around my carboxylic acid. How would you classify a carboxylic acid? As polar or non-polar? And let's think about what constitutes something being polar, or what's a good group for a polar group. We have an OH that usually means something's polar. Yeah, we classify that as polar. And now I'm going to put a box around another group. I'm going to label it number two. How would we classify that OH? Polar or non-polar? Polar. And last but not least, this third box. Well, we've already done it once, so we'll just call this one polar as well. So now, thinking about this entire amino acid, which when we classify amino acids, do we care about the backbone? No. So how would you classify this amino acid? And this isn't a trick question. We got a bunch of groups on that R group that are classified as polar. We classify this entire amino acid as polar because that R group, Chris, doubling down, perfect. Polar negative, I would too. So this is a good question to me of applying what you know. Because this is kind of like, if so long as you understand what polar and non-polar is, then you're going to be able to answer this sort of question without being like, oh, was it the T or the R? Did it have three carbons? Did it have four carbons? This is the sort of question that I like to ask because it's not, did you memorize this? I've got some of those questions where it's just like, recall this information, but lots of the questions that I like to ask are, well, apply that. Okay. Now, with that said, that's all that I've got. I've got office hours starting in about 15 minutes. I think I got to take my dogs out for a minute. Um, but I will be in the office hours link in roughly 15 minutes. Um, so I hope you guys have a great day. Poll Stanley, are you referencing the poll question? I missed the polar negative positive question. Um, I'm gonna go. Sorry, I'm gonna stop my recording and then I'll be here to answer any questions anyone has. And